We're delighted to have Amel Tounsi from MasterCell presenting for us today. After receiving a master's degree at the Université Louis Pasteur in France, Amel obtained her PhD in immunology and cellular biology at the Université Catholique de Louvain in Brussels. She then followed a postdoctoral fellowship at a German cancer research center where she worked on the establishment of in vivo models in mice to study the immune regulatory activity of bone marrow-derived mesenchymal stem cells. After her academic experience, she joined the Research and Development Department of Cardio3 Biosciences, now called Celiad, a Belgian biotech company developing a stem cell therapy for the treatment of chronic heart failure. Amel was involved in process improvement and developed release and potency assays. Her expertise in academia, R&D, and the biotech industry was an asset for MasterCell, where she has worked as development and project manager since August 2014.
Thank you, Freya, for this very nice introduction, and thank you all for joining us at this webinar. Well, during this presentation, as you can see here on the slide, I will be discussing about a project that was held at MasterCell with two other collaborators, Therapies and Terumo. And I will talk about the project where we had to internalize and upscale manufacturing of stem cell therapies using the quantum expansion system. This slide here is to remind you that MasterCell is a public company, and in order to be compliant with some legal aspects, I had to show this message. Before moving to the description of the case study itself, let me just give you a short introduction on MasterCell. MasterCell is a European contract development and manufacturing organization. We are based in Belgium, and our mission is to help our clients, mainly cell therapy companies, move their product faster to the market. And we do so by proposing a one-stop shop company where we can help our clients from the tech transfer service to any development, manufacturing, quality or logistic aspects of their drug development. In order to keep up with our mission and help our clients, we make sure to monitor and adapt to the market challenges. Of course, this is only possible by collaborating with scientific experts that can understand and help the cell therapy biotech companies in their drug development. And last but not least, MasterCell experts in operational know-how are also here to advise our clients on how to optimize their production, mainly according to the GMP requirements. Another key asset of MasterCell, it's the 600 square meter EMEA compliant GMP facilities. You can see here a plan of the facilities, and on the bottom right you can see the tech transfer lab where the work of this case study was performed. You see also that MasterCell has 5 clean rooms, 1 QC lab, and a storage room dedicated to our clients' needs. I was happy to participate in this project as project manager. You can see in this slide that this role is very central, as it is actually an interface between the customer's needs and the MasterCell internal team. We make sure that we actually answer our client's need by gathering the best team internally. As mentioned earlier in this presentation, this work was performed with 2 other partners, Therapies and Terumo. Actually, this was an ideal collaboration, especially for a project where a bioreactor was tested and upscaled. The client wishing to test and optimize the bioreactor were part of the project. The bioreactor manufacturer, Terumo, and ourselves as a CDMO with the GMP know-how and the GMP facility. Therapies and Reagenesis had a well-characterized cell therapy product that needed to be amplified. Terumo was offering a bioreactor, the quantum cell expansion system, a bioreactor based on the hollow fiber technology. MasterCell offered the expertise on GMP cell therapy manufacturing. The purpose of the project was to multiply by 10 the production of adhesive cells. Preliminary work was performed on one quantum, and the strategy was to scale out the production to running 10 quantums in parallel. The average production of one quantum expansion system was of 1 billion cells. Logically, the run of 10 quantums in parallel would need to generate 10 billion cells. The challenge of this 10-time run was the space and footprint of such equipment, the time challenge of harvesting 10 quantums and respecting the cell's shelf life, and training the team to this larger scale and ensure, of course, that all cells generated in the 10 quantums were homogeneous and comparable to the one single quantum run. Concerning the space, as you can see here, the work was performed in our tech transfer lab. This is a 32 square meter room. That is non-classified, but this is not a problem as the quantum system is a closed system. As you can see here in this picture, the 10 quantums were disposed on tables that were 3 meter times 1.5 meter large. They were disposed as 5 quantums back to the other 5 quantums, and the bottles you see on the front are gas bottles and connected to the quantum. In terms of space organization, it's of course important to keep some space around the quantum so that the operators can sample and manipulate the quantums. 2 laminar airflow were placed facing each group of 5 quantum expansion system. From the way the operator is dressed, you can see that this lab is a non-classified lab. But as a quantum expansion system is a closed system, the back connection and sampling can be performed even on a non-classified room. The sterility of the product was not affected, as this was confirmed by the testings performed at the end of the run. I wanted to give you as many practical details on this study, and I think that the insight on the timelines is an important one. The expansion process for each run was of 6 days and is represented in this slide by a blue line. The QC testings were performed after the runs and represented 2 weeks, but you don't see them represented here on the scheme. For all the projects, we had 3 phases. The first one concerned the training and technology transfer that lasted for only 4 weeks. And this was mainly due to the bioreactor manufacturer support, Terumo, in the training, and also to the initial work performed at Regenesis and Adhersis that actually made a pre-optimization of the protocol of amplification of those cells in the quantum expansion system. Once the first 3 training runs were performed on single quantum systems, the QC testing confirmed that the cells generated were equivalent to the client cells, and then we decided to move to the next step, which was the process optimization at MasterCell. The runs in this phase were also performed on single quantums and actually aimed at improving the process. Optimization was mainly performed on culture media flow optimization, culture media consumption, and cell feeding timing. Once the process was optimized, the decision was taken to move to the 10 quantum scale out runs. 2 of them were performed, as you can see here, in the last phase of the project. You notice that the timing between the small scale, the single quantum runs, and the 10 quantum expansion runs in parallel was only done after a week. And I think that's another advantage of choosing scaling out versus scale up. You don't change actually your bioreactor system, so that makes life easier and moving from one step to the other faster. Another important topic in the production of cell products is the workload associated. In this slide, we made the comparison between a single quantum run in the bottom of the scheme and a scaled out times 10 quantum run in the upper part of the scheme. The process, as mentioned previously, is a 6-day process with a day of preparation in order to pre-coat the holofibers of the bioreactor. The time required by each operator was represented by orange lines. And you can see here that in conclusion, the workload associated with the scale out, so if we are multiplying by 10 our production, we're only increasing by 3.5 times the workload. Well, you mainly need more time for the quoting, the daily monitoring. Other than that, the harvest timing had to be kept the same between the single run and the 10 quantum runs. As you know, this is requested by the manufacturer and by the company that the freezing solution and the harvested cells stay at the same time out of the quantums. So therefore, an additional operator was required to keep those tight timing. I also wanted to speak briefly about the harvest day and the downstream pooling steps of the 10 quantum runs. Actually, no specific or additional equipment other than a centrifuge were needed to wash and concentrate the cells. And as the cells, when harvested, needed to be frozen under the same timing as for a single quantum run, which was to harvest the first 4, then the second 4, and then the last 2 quantums together. Once all cells were washed and concentrated, they were pooled and conditioned for freezing.
In this slide, you can see that in both runs, using the 10 quantums in parallel, we managed to generate around 1 billion cells per quantum, just as for the single quantum runs. Proliferation efficiency was not lost. Moreover, the viability of the cell was above the 95%, and the variability in cell yield was of 12%. As mentioned previously, even though the work was done in a non-classified room, we had to prove that the cells were safe, and therefore conducted the usual safety tests at master cell. The sterility test using the bacti-alert method, the mycoplasma test using a validated qPCR method here at master cell, and the endotoxin detection using the LAL chromogenic test. All 3 tests were done according to the European Pharmacopoeia and showed to be negative for all samples. Run 7 was a single quantum run, while run 9 and 10 were 10 quantum runs. This is a proof that the handling of the bag culture and sampling did not affect the closed system and generated cells even in a non-classified area. In order to prove to our client that the scaling out did not affect the cell's characteristic, the usual negative markers were tested for their cells, and they showed to be negative in both single and 10 quantum runs, while their positive markers had the same level of expression on both single and 10 quantum runs, again validating here the comparability between both runs. Another important parameter to show the comparability of the cells generated from a single and a 10 quantum runs was the potency of the cells. Atasis considered their cells as potent if they secreted a certain number of proteins, and Regenesis performed those testings and confirmed that the potency of the cells was equivalent in both single and 10 quantum run equivalent. In this slide, I wanted to summarize the conclusion made during this project. Well, first of all, we were very surprised by the ease of manipulation of such a bioreactor. The operators involved in the project have never worked with a quantum before, and could easily learn and operate independently the bioreactor by the end of the training period, which was already after a month. This, of course, led to a significant decrease in the project timelines, and therefore we could run all development, tech transfer, and 10 quantum runs in just 16 weeks. On the other hand, we also fulfilled our challenge to increase the cell expansion capacity of our client by about 900% in reference to a single quantum run. The comparability study showed that the cells generated after scale-out were equivalent in terms of safety, yield, and potency to the single quantum runs. Another important element concerns the workload. It is interesting to note that by increasing the labor by only 3.5 times, we could multiply by 10 the cell production. And I also would like to remind you that all the work was done in a 32 square meter non-classified tech transfer lab. It's an important information for a manufacturing organization, as clean room footprint is an essential parameter in the cost of cell therapy production. The other advantage we also have seen in this project is that the scale out of the process did not modify the process length and maintained it at 6 days. The training of the team actually to move from a small single quantum production scale to a 10 quantum scale out manufacturing scale actually was not required, as they were actually ready to go from one system to the other. I wanted to close this presentation by thanking all the people that were involved in this project. I remind here that we worked with several collaborators, Therapies and their company here in Belgium, Regenesis, as well as Terumo BCT, and I really would like to thank all of them for their help and support throughout the project. I hope that you all enjoyed this presentation. I wanted to give you some practical insight, on the scale out of manufacturing cell therapy and moving from one bioreactor to running 10 in parallel, with the challenges that it takes. Of course, I am now ready to answer all your questions. If you need any further information on MasterCell, you can also contact us.
Thank you, Dr. Tounsi, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. Your questions will be answered today by Dr. Wenyan Leong, Cell Processing Specialist, Asia Pacific at Terumo BCT. Wenyan Leong, PhD, is the Regional Specialist for the Quantum Cell Expansion System, responsible for managing knowledge transfer and implementation of efficient and safe manufacturing solutions in the biotechnology cell therapy field. Dr. Leong works very closely with various industry representatives, researchers, and regulatory agencies in Asia Pacific. She is passionate about the advancements in cell-based therapy and seeks active dialogue with stakeholders in the field on how to bring research ideas into the industry. Prior to joining the cell processing team of Terumo BCT. She spent 4 productive years in academic research on cell-based regenerative medicine platforms, working on a variety of biological cells and biomaterials. Her contribution of 10 research articles and 3 patents to this specialty resulted in a PhD, bioengineering, conferment from Nanyang Technological University, Singapore. Now let's get to our questions. Our first question is, are you able to provide an estimate of cost of goods for producing 1 billion cells? Did this scale out linearly for the 10 times manufacturing scale?
Thank you for your question. We have various customer sites who have analyzed the cost of goods for quantum versus their manual processes. We are unable to provide a universal estimate because of the variability in process and protocol across different sites and countries. When manufacturing is scaled up to several quantum systems, the cost of goods is expected not to scale out linearly due to the non-linear increase in infrastructure, space, and labor. In the Therapies, Regenesis, and master cell study, where they scaled out from 1 quantum to 10 quantum systems, there was no need to increase space and team of 2 operators. Labor hours-wise, there was a 3.5 times increase operating 10 quantum systems compared to a single quantum. The study also reported a 40% reduction in cost of goods relative to manual manufacturing methods. This cost of goods observation is quite consistent with those mentioned in other sites, for example, Mayo Clinic and Catapult.
Thank you. The next question is, what protein do you use for coating and at what concentration?
We use 5 milligrams of fibronectin to coat the bioreactor.
Thank you. Have any studies been performed in the quantum to induce differentiation in the bioreactor in situ, for example, hepatic?
There has been some work on in situ differentiation in the quantum cell expansion system. However, these are customer sites and we are not able to disclose any specifics.
Thank you. What cell dissociation reagent is used for harvest?
In most of our work with customers, we use whatever reagent the customer has been using. Most will use 0.25% trypsin with EDTA.
Thank you. I noticed that you hang 5-liter bags on the bioreactor. How many days does a 5-liter bag go through? Do you keep the bags at room temperature with no protection from light?
Thank you for your question. So the media bags are 4 liters in capacity. The quantum cell expansion system is a perfusion-based system, which means there is a continuous feed rate and waste removal. How long a bag can last, therefore, depends on the feed rate, which is adjusted throughout the culture to provide the cells sufficient nutrients. Typically, for a 7-day culture of adult mesenchymal stem cells of passage 1 and above, we use 6 to 8 liters of media to obtain 250 to 750 million cells. The bags are kept at room temperature with no protection from light. However, if there are sensitive media components, we do protect these bags from light by wrapping them in tin foil.
Thank you. Is the harvest time exactly 6 days from plating the cells into the reactor?
The duration of culture depends on the cell type and several other factors, including passage number, doubling time, and media components. Ultimately, it depends on the cell growth curve.
The end goal is to achieve maximum number of cells in a healthy state. Therefore, we aim to harvest before the cell growth curve reaches the stationary phase.
The next question is, I know the quantum gives you the option to use protein-free media in the extracapillary loop and complete media in the intracapillary loop in order to save on media. Did you try this capability?
Yes, in some of our customer sites, we have performed a dual media feed strategy.
Thank you. The next question is, did you load the cells using ROCK inhibitor?
No, there has not been a request to. When we work with customers on transferring the process from manual to the quantum cell expansion system, we try our best to follow the manual protocol in terms of the reagents used.
Thank you. We have time for one last question, and that question is, did you find it necessary to change the composition of the cell culture media from a normal culture to the hollow fiber system?
Thank you for your question. No, we have not found a need to change the composition of the cell culture media customers have been using.
I would like to once again thank Dr. Tounsi for her presentation and Dr. Leong for answering questions. I would also like to thank LabRoots and Terumo BCT for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through March 12, 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.